All right, excellent. And closing out our session, we're going to get a little bit more basic with a research talk by Dr. Cynthia St. Hilaire. She's going to talk to us about from the bench to the bedside, novel druggable target driving in aortic stenosis. Dr. St. Hilaire.
Thank you. Thank you for the opportunity to be here. I guess I'm definitely switching gears. So I do have a disclosure. We're patenting some of this technology. But my lab, I have a lab at University of Pittsburgh in the Department of Medicine and Cardiology, and we do basic and translational research. We are very much interested in the biological mechanisms driving calcification diseases, and specific to this meeting today, calcific aortic valve disease. And so, it really wasn't until 1993 when Linda Deemer and Christina Bostrom at UCLA really defined that calcification in the vasculature, whether it's in the valve or in the vessels, is an active biological process. It is not just the breakdown of the tissue. And we in my lab are very interested in understanding that process that causes a healthy vascular cell to transition into a calcifying cell. And our main questions are stemming around or swimming around this idea that is it maybe a one or two step process where just a couple genes or a couple proteins are dysregulated, or do the cells actually undergo a very complex transdifferentiation, where they maybe assume, stem cell-like properties, and then undergo a transition that makes them very much osteoblast-like cells. And so as we were thinking about calcific aortic valve disease, because the other diseases I'm working on are genetic-based, we really went back to the tissue, the cell source of a lot of the cells in our tissue. And in a lot of the soft tissue, including the vasculature, it's the mesenchymal stem cell. And as we're digging around, we found these really interesting studies. These are rather old. They're from 2002. And when they overexpressed this gene called, TERT, or telomerase, which hopefully everyone in the audience knows is what helps keep a cell healthy. It extends the telomeres of a cell. And that overexpression didn't immortalize the cells. It actually drove them to calcify. And so I'm not going to give you a big biochemical talk. I know you're all physicians, but suffice it to say that under normal conditions, this gene TERT helps to elongate the telomeres at the end of our chromosome, but a lot of recent and really interesting molecular work has identified that it's actually also a chaperone that can help harness the chromatin remodeling components that help a cell transition between its fate. And so we started to ask this question, I think it was in about 2017, does overexpression of TERT help drive the osteogenic transition of cells? And we're very interested in this, obviously, because if we know what's driving it, we can perhaps prevent it from happening. And so that's when I started collaborating with cardiothoracic surgeons here at UPMC in the HVI, and this lower valve actually we got from Eames. And we started with a tissue-based approach. And so looking at these human tissues, we started to measure this protein in the tissue and found that it was very up, as you can see in the pink here. I don't think I have a pointer, but it was very high in the calcified tissue. And we were able to develop a very unique protocol, well, not very unique, but hadn't existed before, where we can actually collect cells directly from these patient valves, but also from donor cadaveric tissue that is up to 2 days old. And we can process these cells in special solutions that allows them to survive for in vitro disease modeling. And so we can actually get cells to calcify in a dish. That's what you see with this red stain. And we started to do kind of basic molecular biology approaches to understand the contribution and the requirement of this gene TERT in our cells. And if we were overexpressing it in the valve interstitial cells that we would collect from patients, we could prevent calcification in a dish. And this is a standard, in vitro disease model used in the field. And when we wanted to think about what could be driving this, what could be driving this protein TERT to be upregulated, it really shouldn't be there if a cell is not dividing. We went back and started to look at some of the cases where you get early calcification of the valve. You know, things like inflammation and interferonopathies drive some of that pathology. And there's even some studies in vitro using porcine cells, I believe, that showed that interferon signaling can help drive the transdifferentiation of a valve interstitial cell. And so we started to ask that question and looked at this protein RUNX2, which is required for a cell to calcify. And so this RUNX2 is highly up in our cells. It is present in the nucleus when these valve interstitial cells are undergoing osteogenic transition. And we can do these molecular assays called proximity ligation assays, where we can actually see TERT and STAT co-localization in these valve interstitial cells in our in vitro disease model. We can also show binding through immunoprecipitation assays, and also some more direct and also unbiased approaches that are showing this complex of TERT and STAT5 are honing in to the RUNX2 sites in the DNA, helping to drive the expression of this osteogenic gene RUNX2. What we've then wanted to do is try to leverage this to prevent calcification. So if we use an inhibitor of telomerase, our cells will essentially age, right? We can't shorten our telomeres. Also, a telomere inhibiting drug doesn't prevent calcification. We're talking about the non-canonical effects of TERT. So we went down the other pathway, which is STAT5. And if we use known available inhibitors of STAT5, we can prevent calcification in addition. So this is showing you data from both human valve interstitial cells, but also coronary artery smooth muscle cells. We also then go back to the patient tissue, and we have found that TERT and STAT are co-localizing in the nucleus of cells of calcified valve tissue. Really importantly, where could we actually try to target these pathways for therapies is perhaps in a bioprosthetic valve, right? If we could perhaps coat a bioprosthetic valve with a novel molecule to prevent the osteogenic transition of cells, that might expand the lifespan beyond the 7 years. And so we're actually seeing that the cells on top of and kind of embedded in bioprosthetic valves are also showing these signatures of TERT and STAT5 in their osteogenic, as these cells are calcifying. You can see them in the green and the red on this image. And so to kind of summarize our findings, we think that this initial inflammatory insult is driving the activation of these genes in the valve interstitial cells, and they're coming together and activating this osteogenic transcriptional program that is driving the transdifferentiation of a valve interstitial cell from a healthy to a calcified state. We're now really working on trying to leverage and understand what's happening, how this is working, and so we've taken kind of a cool AI approach where we can start to model these using AlphaFold and identify how exactly these proteins are interacting. We're also developing tools to watch this in our cells in live time using molecules that can be attached to our proteins of interest, our TERT and STAT5, and so we can see in a dish in a large format system when and where these are interacting, and also then screen novel and established biological compounds to prevent their interaction. And so that was, I've never given a short 8-minute talk before, but that was a successful one. So this obviously takes a massive team to do, not only with the clinical side of things, but my lab. This project was started by a former postdoc, Luis Hortels, who's now a PI in Germany, and really spearheaded now by Rolando Cuervas. But of course, it takes a village and wonderful collaborations with HVI and my favorite surgeon, Ebes. So thank you.